Thanks very much, Brian. Gray had the really difficult job tonight because he had to give you all the boring details of everything, but Gray's also funnier, so I think he kind of made up for it. I'm now going to show you some real-life examples of how we put some of these techniques into use and why sometimes the perfect sampling regime may not be the best one, depending on resource constraints, time constraints, everything else. A big question that we're all interested in, so how is climate change impacting marine biodiversity? Well, I think first, what do we mean by climate change? When I first started 15 years ago, climate change just meant temperature. That was the big driver we were all looking at. Today, I think as marine scientists, we realise there are far more variables linked to anthropogenic CO2 influences on the planet. Has anybody read the IPCC Fifth Assessment Report? Don't. Read the Summary for Policymakers. There's a fantastic set of working group reports, but they're over 2,000 pages each. So unless you're really interested in the details for the physical oceanography or whatever, the Summary for Policymakers is really short, it's really good, and it's got some really nice graphics. What it essentially says is it's increased its confidence from the fourth assessment report. Nothing much has changed in terms of how we think the Earth is warming. There's quite convincing evidence, I think, in changes in the global ocean average upper ocean heat content there, the top graph, and there's high confidence that this change in warming is being driven by human beings. The great thing now is that we also have ocean acidification firmly implanted in the IPCC, and there's a chapter devoted to it. This is also a potentially really important driver because, as you know, the oceans are a major sink for atmospheric CO2. That changes the pH and the acidity of the oceans, and this may well impact in different ways to temperature. It may be antagonistic, it may be synergistic, or it may just be additive, but we have no idea yet. So it's another important driver. And you can see on this bottom graph here, we've got surface ocean, there's CO2 and pH, and this is time along the bottom. And you can see that there's been a huge decrease in the pH in units relative to organisms over this time. So there is a real and discernible change in the ocean chemistry as well. So what biological data do we use to try and work out if these are having any impacts? This is a massive plug for a recent paper. I'd like you all to read it, cite it. It's fantastic. The UK has got some of the best and the longest time series in the world, both biologically and also for physical oceanography as well. We've just had a special edition come out in Philosophical Transactions of the Royal Society. My one talks about marine biodiversity, but there's all sorts of different papers. And these chart the introduction of time series, why they were first started, not all question-driven in those days. Sometimes they used different methodologies. Gear use, for example, for the fisheries has changed with time. So when you're looking at time series data, as Gray said, methodology is very important. But we've got to remember, in the late 1800s, people didn't understand replication because it didn't exist. So sometimes the methodologies we started with are not always as scientifically robust as we would use now. So we have to look at ways we can continue the time series in a way they're comparable, but also maybe make some adjustments to account for the modern statistical analysis as well. There's just a couple of examples here that aren't mine. On the left-hand side, these panels here, this is a continuous plankton recorder that looks at phytoplankton and zooplankton across the northeast Atlantic. And what it's shown here, the left-hand panel here, one of the more warmer water affinity species of calanoid copiapod. What you can see here is in the 50s to the 80s, they were mainly situated off the south coast of the UK. But what they've done up into the 2000s, with warming of the ocean temperatures, these species that prefer these warmer waters have actually followed the temperatures north. These are the subarctic species that prefer colder waters, and again they've moved north. The problem here is they've actually moved out of the North Sea, as you can see coming down the time series here. This has had implications further up the food chain. Atlantic cod is a major source of fish produce in the UK, and the cod feed on the cold water copiapod. They tend not to seem to feed on the warm water congener. So the plankton move with the warming temperatures, and the fish have had to move with it because their food resources changed. There's been less done on the physiology of cod, but it's also like that the cod, because they're mobile, are moving themselves to try and keep themselves in their suitable climate space as well. So these are just a couple of examples of how long-term time series have really been able to show us what happens with climate change. how do we collect relevant, and I think these days robust data across large spatial scales? So if you're Tony Underwood and you went to the same place for 20 years in this little bay you did in Australia, that's great because you can throw out as many quadrats as you want, replicate like crazy. But that technique's not going to work if you're going to try and sample, for instance, the whole of the Northeast Atlantic. Now, the project that I run, the Marklim project, looks at marine biodiversity of rocky intertidal habitats and climate change. This is a UK-wide project that was started in 2001 when I was actually a brand new PhD student and the dedicated research assistant on this. I was supposed to be 50-50. After 2 weeks, Professor Hawkins, who was my boss, my director and my PhD supervisor, decided that I needed to work 100% on this and the PhD could happen on Saturdays, Sundays, evenings. So I was fully committed to this. And what we did with Marklim is we took historical data collected by some really insightful people, Professor Alan Southwood, based at the MBA in Plymouth, Professor Dennis Crisp, based at Bangor University in Wales, and Professor Jack Lewis, who was based at the Robin Hood's Marine Laboratory up in the North Sea. Now, these guys recognised very, very early on in the 40s and 50s that when the sea temperature changed, we had fluctuations between pilchards and mackerel, so different types of fish, the plankton changed, and they saw these oscillating cycles. Before anthropogenic climate change, they could track different climatological, like sunspot cycles and that, but they realised that, that marine animals were very, very good at tracking changes in the temperature. So we took all their raw data, which was in notebook form, and used this as a baseline. We then went and repeated the classic broad-scale surveys at their sites, and we established a long-term network of sites to continue to monitor change every year from 2001. We used to forecast future change. The best scenarios until very recently come at the regional and national level from the UK Climate Impacts Partnership. So that's the Hadley Centre that do all the modelling, and the Met Office, which give us all our meteorological and climate data. So this would be very similar to kind of NOAA and NASA data sets that you work with over here. It's a very important part of the Marine Biological Association's history, and also for myself personally, that we can use this data to inform policies about conservation, about planning, and address the implications for society as well. Fantastic blue skies research so often just gets published and put away in a journal and never used again. And I think it's really important, if you do something that's great and you can show an applied use to help the environment or to help people, then why not? So this is my project, Marklim. It's the most spatio-temporally extensive intertidal data set globally. And we've also been able to show from this some of the fastest biogeographic range shifts in response to climate in any system. This picture on the left here, this shows our survey sites in the UK. I've also now set up long-term networks in France and Norway as well to increase the biogeographic coverage. And if you look where the arrows are, these show some of the hotspots for range extensions, so the leading edge at high latitudes. And then we've also got some of our native boreal species that prefer cooler waters, and these arrows show where the trailing edge of the ranges are disappearing. Now, if you look at the next image here, this is just a snapshot of sea surface temperature, but it also shows you again not only the gradients that we have around the UK, but the UK sits across this major marine biogeographic boundary between what we call the boreal cold waters to the north and the Lusitanian warm waters to the south. So we get many species that are of a similar type. So we have many species of limpets. Some are boreal in their distributions and reach their southern limits in the UK. Some are Lusitanian and reach their northern limits. And in this area, a lot of them are overlapping and competing for space. So not only can we see how individual ranges shift, we can also see how climate change is making one species that's functionally very similar react very differently to the other. We also have wave fetch here. This is that panel. With climate change, we're predicted to get higher wind speeds, greater waves, impact and more storminess. And with the rocky shore, again, this may well affect which species are able to live where. And this end panel here represents quite a recent development for the marine environment, but something that seems to be working very well, and that's the velocity of climate change. So it's not just looking at a snapshot or an average from the 60s to the 90s and then onwards. What this actually looks at is it tracks how fast in each of these little cells the temperature has been changing. So it's now allowing us to do more dynamic modelling of species distributions as well. Methodology. It has to come back to that, I'm afraid, but it's not going to be a really, really long one because Gray's covered it. So what we did is we used the same methodology for the original surveys. If we changed it, we'd have lost a time series to compare it to. And the first year, we surveyed over 400 sites that we'd taken out of the old data sets to see which ones were suitable, good for the species we were looking at. And we managed to trim this down to something manageable. So we do about 100 sites around the UK annually now. We can't manage 400, unfortunately. And again, that's where Gray says, you know, you can work out your optimum, but you've actually got to be realistic in terms of times and resources as well. An important thing we did is institute sites beyond the range edges, because we wanted to see if, since the time series, which most of them were stopped in the 1980s, had anything changed, so had things moved beyond those range limits by the time we came back to restart the time series again. A very important other point as well, especially dealing with time series, but if you want to compare movement of anything, is to record absences. A big problem if you don't record absences is you don't know whether the species was there or not in the past. It's like if you go to museum record collections, it's the same problem. If a species doesn't exist in the collection, does it just mean it wasn't at that location, or does it just mean that they weren't looking for it? Maybe it wasn't of interest to them then. Invasive species, this is also really important. Again, if you have an invasive species that arrives in a man-made habitat, a marina, a port or something, if you can record absences around that introduction site, you can then also start to record the spread, the rate, the nature and the pattern. So absence is as much important as detecting presence. Before we even start the survey, this is some of the most important information that you can collect. And we had to go back through the archives of the time series, and we really struggled with some of it, trying to work out exactly where the sites were. So we've refined it now. We have a standard site name. It's always the same for that location every year. We also put the grid reference. In the UK, some of our old maps were done with set grid references before we had latitude and longitude. So our 50s, 60s, 70s data has grid references. We still use these maps, but now we also use the lat and long, the access point, and the lat long and the centre of the survey area as well, so that in the future people can get right back to the same spot. One time we went up to North Scotland to Far Point, and we followed where the grid reference was and the site, and got to the top of a 100-foot cliff and thought, yeah, that's probably not, it's probably not where we're supposed to be. We also put down who was the recorder or the recorders for the survey, so which people did it. And again, Greg talked about inter-surveyor error and bias. Markham has only ever had a very, very few number of surveyors. So Professor Alan Southwood trained Professor Hawkins and myself, and Professor Hawkins and myself have trained the other 3 people that work with us on the surveys. But basically we've got a very, very small team dating back to the 50s. Everybody is intercalibrated with each other, so we all do the same things at the same time, and we cross-calibrated. So we're very, very confident that we have low, if not zero, operator error. Weather conditions, that's really important. Sometimes the weather means you can't actually survey the area as properly as you'd like to. Visibility, well, I guess it's not so bad here, it may be a bit more like the UK. Sometimes it's thick fog or thick rain, or it's snowing, and then again this may well affect your results. So when you go back and find an anomalous point, you may be able to go back and work out that it was just because we couldn't do the survey properly. Low shore availability, again, if you've got a massive swell coming in, we can't survey the kelps possibly as quickly as we could. So this is really important to get the survey metadata down first. And the methodology. Now we're stuck with the categorical abundance scale that was adapted from Crispin Southwood in 1958. Like I said, if we changed the way we did things, we wouldn't be able to compare anymore. And I'll come on to how we do this shortly. But what we also decided to do as well was to look at replicating. The top shells that we work on, Jack Lewis and colleagues worked on these in the 1980s. Now they did do different counts, but again, replication wasn't a big deal in ecology then. So they used to grab a few handfuls, measure them all on occasion. The last year of this time series in 1986 was the one and only time they did replicated time searches. So we have one time point that we can compare to statistically. The reason we do the counts there instead of putting quadrats down is these species don't live on flat rock. They live in crevices, under cobbles and boulders and everywhere. So if you can imagine you put this standard unit down, because we use the 50 by 50s, if you put this down on a flat surface and then you put it down over a lot of cobbles, boulders, cracks, then you're not actually doing what this is set out to do, which is to give a standard area in which to survey. So So we don't standardise by area for these species, we standardise by time. So we grab a bag, and it's a bit like a trolley dash. You literally get on your hands and knees and you put as many of that species in as you can in 3 minutes. Bag it up, seal it, do it again 5 times. So we have replicates by time instead of space. Then for the limpets and the barnacles, because they do tend to live on flatter surfaces, we use different size quadrats. Again, it's about selecting the size. The limpets we do 50 by 50 centimetres. The barnacles, we do a 5 by 5 centimetre because I am not counting all the barnacles in a 50 by 50. I think we worked out that over the years, I've personally counted by hand over 500,000 barnacles as it is, so that's sufficient.